Welcome to our video for chapter 25, and this is only the first part. There's going to be two videos that I'm going to be doing for chapter 25. The class is progressing very well, so we're almost halfway into human anatomy 2. We're now going to get into the urinary system. Let's begin. So the urinary system is actually the it's actually the major excretory system of the body. And it maintains the body's internal environment by regulating the total water volume and total solute concentration in the water itself. It is also regulating ion concentrations in the extracellular fluids. It ensures long-term acid-base balance, excreting metabolic wastes, toxins, drugs, and producing erythropoietin as well. You remember erythropoietin is produced by the liver. This one regulates blood pressure. And it also is responsible for the production of a special hormone, which we are going to start uh, learning today. And we are going to still continue to learn that when we get into the endocrine system. So renin is actually the one that regulates red blood cell production and blood pressure. It has something to do with blood pressure. It also is responsible for activating vitamin D, carrying out gluconeogenesis if needed, and the kidneys are part of the urinary system. This is actually a system which also includes the ureter. That's actually the ureter right there. Let's take a look at that. That's the ureter right there. Okay, that's the ureter. It transports urine that we produce in the kidneys, from the kidneys to the urinary bladder. And the bladder is only a temporary storage and reservoir for urine. And once it gets filled up, then we actually eliminate the urine. The urethra would transport the urine out of the body from the bladder. Okay, we're going to start with the gross anatomy of the kidneys for this particular video. And let's take a look at the location of the kidney. Kidneys are retroperitoneal, and you could see it from this picture right here. They are located in the superior lumbar region, which is located between T3. Okay, this is actually already uh, the 12th thoracic rib, so it is already between uh, T, not T12 and L5. That's actually the location of the kidney. The right kidney, though, is crowded by the liver and therefore is a little bit lower than the left. The adrenal gland sits on top of each kidney. Now, the kidney has a peculiar shape, and I don't know which one is actually which word came in first in the development of our language. Is it the beans, or is it the bean shape, or is it the kidney shape? So we say the kidney is shaped like a bean, but when we're looking at the beans, we say that the beans are shaped like the kidney. So they do have the same shape. They are convex lateral. They have a convex. They both have convex lateral surface and a concave medial surface. The only difference with that is the kidney belongs to a living body, and the beans is actually a plant. Okay, so or a part of the plant. So in the concave medial surface, it would pretty much like carry what you call the vertical, renal hilum, and it leads into the internal space of a kidney, the renal sinus, and the ureters, the blood vessels, the lymphatics, and the nerves would enter and exit at the hilum of the liver of the kidney. The upper parts of both kidneys are protected by thoracic cage. You could see that right here. And The perirenal fat provides cushioning and support so that the kidneys are not going to drop. As a matter of fact, if a person becomes anorexic and too thin, and it will lose the perirenal fat, the kidneys may drop and it might kink, and the ureter might kink and would restrict the flow of urine for elimination, and that could pretty much like destroy the kidneys themselves. The lower parts of the kidneys are susceptible to blunt trauma because it's not protected by our rib cage, especially the right kidney, okay, which is a little bit lower. So falls such as motor vehicle accidents also, uh, falls and some contact sports injuries will hurt our kidneys, and most especially the right kidney. The renal artery is especially vulnerable to injury from rapid deceleration during car crashes. It leads to lacerations, okay, that means what do you call this, blood vessels, or thrombosis, blood clots. Hematuria or blood in the urine is an important sign of such trauma. So in this case, surgical treatment may be required. Okay, so there are three layers of supportive tissue that surrounds the kidney. The first one is what you call the renal fascia. Okay, these are the supportive tissues of the kidney. You have the renal fascia, you have the perirenal fat capsule, and the fibrous capsule. So the renal fascia is actually this white stuff right there, which is pretty much like already part of the peritoneum. And the perirenal is actually the fat capsule that surrounds the kidney, and then there's a fibrous capsule that surrounds the kidney as part of its cover. So the renal fascia anchors the outer layer of the dense fibers and is made up of dense fibrous connective tissue. The perirenal fat capsule is our fatty cushion. Your fibrous capsule is a transparent capsule that prevents the spread of infection to the kidney itself. The internal kidney has three distinct regions. This is actually a diagram of the kidney. This is a real human cadaver kidney. Okay, so the renal, I am going to pretty much like concentrate on the discussion by pointing on to the diagram instead of the dissected cadaver kidney, because we never really dissect cadavers in AMP2, in college AMP2. This would happen if you are going to get into med school. So first of all, let's like take a look at the distinct regions of the kidney. First, you have your renal cortex. This renal cortex is a granular appearing superficial region. So this entire thing is your renal cortex. Then you have your renal medulla, which is actually deep into the cortex and is made up of cone-shaped medullary renal pyramids. So the only thing that looks like pyramids will be these guys here. And therefore, this section right here is actually your renal medulla. Now, because of the shape of the kidney, then the pyramid, the broad the broad face of the pyramid usually faces the cortex. Then you have the papilla, which is at the tip of the pyramid, right there, the tip of the pyramid. Pretty much these points internally, okay, right, going into the hilus itself. The renal pyramids are separated by renal columns, and these are your renal columns. They are pretty much inward extensions of your cortical tissue. So you have cortex here, sort of like an inward extension of the cortex is actually your renal columns. And you will find a lot of blood vessels in the renal columns. The lobe pretty much is when the medullary cavity and its surrounding is actually made up of the medullary pyramid, which is actually this one, and its surrounding cortical tissue. About 8 lobes per kidney. So 8 lobes of this is actually present in each kidney. The renal pelvis is actually this area right here. That's your renal pelvis. This is a funnel-shaped tube continuous with the ureter. So you could actually see this, and it's continuous with the ureter. That's your ureter right there. And you have also the minor calyces. The minor calyces are cup-shaped areas, and the minor calyces are right in that section right there. That's your minor calyces. These are the cup-shaped uh, areas that collect urine. So your um, your nephrons actually would empty urine into this area right here. And the papilla, which is actually this particular cup-shaped region right here with holes in it, uh, pretty much would empty or drain the urine into the minor callus. So, okay, so the minor callus together will join a bigger tube, which is called the major calluses, and all the major calluses or all the calluses actually empty into the renal pelvis and then into the ureter. So the major calluses, which is actually the joined areas of the minor calluses, collect the urine from the minor calluses. They empty their urine into the renal pelvis, eventually flows into the from the pyramids into the minor. Okay, the urine flow. Let's take a look at where the urine flow. Okay, the urine flows from the nephron. It's now going to flow into the renal pyramids. Okay, the pyramids itself right there, into the pyramids. The pyramids is now going to empty the urine into the minor calyces, into the major calyces, into the renal pelvis, that's your pelvis right here, into the ureter, then from the ureter down into it will be temporarily stored into your bladder, and then from the bladder into the urethra to the outside when you void. All right, let's take a look at some homeostatic imbalances. One which we call pyelitis is actually infection of the renal pelvis. This is actually the renal pelvis itself. And the other one is pyelonephritis, which is actually what this is. Pyelonephritis is the infection or inflammation of the entire kidney, and the infections, most especially among females, because we have a very short urethra. The infections or inflammation is brought about by um, by a bacteria that can actually go through that short channel and pretty much like find their way into the kidney, and it causes severe swelling of the kidney and abscess formation. Usually, it is not without hope. This could actually be reversed. The inflammation can be reversed. It can heal with the help of antibiotics. So the treatment is usually antibiotics. And most of the time, if it is actually like treated on time, it is going to be successful. So now let's take a look at the blood vessels inside the kidney. Make sure that we know this. Our arteries will be delivering blood away from the heart. So your first big artery is the aorta. The aorta goes all the way down into the abdominal cavity as the abdominal aorta. And the abdominal and the renal artery right here is one of the tributaries of the abdominal aorta. So from the aorta into the renal artery, the blood that has not been filtered yet, okay, is actually your arterial blood. Arterial blood goes into the kidneys, and it will be ready for filtration. So when you are producing urine, you are filtering the blood, and this is exactly where you're getting your urine from the water in the blood, from the waste matter in the blood, which is excreted through the urine, and they go through the kidneys and they go through the process of filtration within the glomerulus and within nephron, the nephrons of the kidney, which we will be discussing later on. So from your aorta right here, okay, you have your renal artery. Then you get into the segmental artery. Okay, then you have the interlobar. The interlobal goes in between the lobes. Okay, then you have the arcuate. You know that the arcuate, this section here, is actually is already running alongside the, what do you call this, alongside the cortex and the medulla. So your arcuate goes into an arch, this arch-like in shape. So, but it's because the top of the renal pyramid is actually it's like a pyramid, and the base would be like an arch. So this particular arcuate arteries is located alongside the margin of the cortex and the medulla. So if you're going to have the chance really to get into directed study and get hold of a real kidney model, then you would know that the arcuate arteries and veins really run alongside, and that's going to be your indication of where the cortex and the medulla is, because that's going to be the margin between the cortex, that's the cortical region of the kidney, and the medulla, and your arcuate arteries and veins are around this side. So from the arcuate artery, then you have the radiating. Can you see that? These radiating, the cortical radiate arteries are radiating right into the cortex themselves. Then from these radiating arteries will be your afferent arteriole, which you will not be able to see here because of the size of the model. The afferent arteriole is the one that gets into the glomerulus of the kidney, and then you have the glomerular capillaries. This is where filtration happens. This is where your blood goes through and will pretty much be filtered. And so the filtrate now will be again further filtered into being reabsorbed and secreted right along the tubes, or the tubular reabsorption and secretion that is happening in the nephron. So from there, okay, from there, the finished product, urine, will go into the eventually into the calyces, and then into the be into the bladder before we actually void it. Now the blood returns to the blood that's been filtered off of what do you call this, of the toxic substances that we have to eliminate through our urine. It's now going to go back and will be exiting the glomerulus through the efferent arteriole. So remember, when you say afferent, this is where it enters, and efferent is where it exits. But this time we're talking about the blood. Okay, so now the blood goes into two major types of capillaries, which we are going to anatomically, which we're going to discuss later on. So it could be that they're going into either the peritubular capillaries or the vasa recta. Then from there, it now goes into again, the cortical radiate vein, then into the arcuate. You see the arcuate veins. Then from the arcuate veins, you have the interlobar. This is a better view of the interlobar veins, okay? And then the renal vein is actually right there. Remember that it does not look like we have the segmental vein. Everything is almost like the same between an artery, the name of the artery, and the name of the vein. It doesn't look like it doesn't look like we have segmental vein. You have segmental artery here, but you don't have any segmental vein. But that is fine, okay? So it's only one of the branches that the veins don't have. So your Renal arteries deliver about one fourth, about 1,200 ml of cardiac output to the kidneys every minute. Wow, 1,200 ml of blood every minute to the kidneys. The kidneys is one of the most hard-working organ in the body. Well, all of our organs are hard-working. You know, we just mentioned in the digestive system how hard-working, what it calls metabolically our liver is, and how helpful it is in detoxifying in the detoxification of the blood itself. Now, here comes the action or the fact that another organ is actually cleansing our blood, which is actually our kidney. 1,200 ml of cardiac output goes to the kidney each minute. Okay, so I mentioned to you already the flow, and there is no segmental vein. Make sure that you know that. And the nerve supply, forgot about the nerve supply. The nerve supply via the sympathetic fibers is actually coming from the renal plexus. So here now is going into, if you're going to get into, can you see that little guy there? That little guy there is actually your, um, this big picture right here. This big picture here has been enlarged, which is uh, a nephron. Trying to take a look at the nephron, and we're trying to position the nephron right there in that little spot right there. So this is your arcuate arteries and veins. And so if this is, I said, these are the margins. They act as the margin. They are the marginal arteries that would be right around the margin of the cortex and the medulla. So this section here is your cortex, and this section here is your medulla. So your nephron, there you go, you have a nephron. It's an odd looking nephron. When I was studying this, when I was a student, I had, my undergraduate was a bachelor's of science in zoology. I majored in marine biology though, but we have a lot of what do you call this, limnology, the study of the invertebrates. And I remember what do you call this, um, worms, tapeworms, okay, would look like that. And they are really nasty worms. Okay, so but these are not tapeworms, they are microscopic, they're very small, and they are our nephrons with the head or the capsule. Okay, it's made up of the capsule and the tubules. Okay, so the capsule part is actually right on the cortex, and the tubules, depending on, well, almost all of the tubules are actually on the, also on the cortex, with the exception of your loop of Henle, which pretty much gets into the medulla. Okay, these are the ones that would concentrate your urine. There's going to be absorption and reabsorption. Fantastic movement right there in the loop of Henle. So your nephrons are, this nephron thing here is actually the structural and functional units that form urine in our kidneys. There's about 1 million nephrons in a kidney. And just like what I mentioned, it has the Bowman's capsule or the renal capsule, and it will have also the renal tubules. And there are two parts to the renal capsule. The renal capsule is made up of the glomerulus, which is actually, if you can see it right through that capsule itself, you see a tuft of capillary inside it, is actually your glomerulus. It's made up of fenestrated endothelium, and then these capillaries, the capillaries of the glomerulus, is highly porous, and that means it allows for efficient filtration. And the filtrate is the plasma-derived fluid that the renal tubules would process. So the plasma-derived fluid, the renal tubules would, the filtrate would be processed through absorption and selective absorption and secretion process and to form the urine. So the glomerular capsule, this entire portion here that holds the glomerulus, is also known as the Bowman's capsule. I've been calling it Bowman's capsule because that's how I learned it from some 40 years ago. It is cup-shaped. It is a hollow structure surrounding the glomerulus and is made up, this glomerulus here, this Bowman's, rather not glomerulus, but Bowman's capsule here is made up of two layers. That it is actually made up of simple squamous epithelium. You could see how fragile it is, but it is actually made up of the parietal outer portion of simple squamous epithelium and an inner visceral layer. And this inner visceral layer clings to the glomerular capillaries, and these are actually um, pretty much like it will differentiate, okay, from a simple squamous. Some of them will form like extensions, okay, and you call them now the epithelial podocytes. So podocytes, pretty much the extensions of the podocyte, um, are like food processes that cling to the basement membrane, forming filtration slits between the food in between the food processes that allows as a it is actually allowing as a what do you call this, a secondary filtering mechanism. Okay, so instead of just the fenestrations, you have another filtration slip in between the processes, the pedicels of your podocytes, and it allows filtrate to pass into the capsular space, and pretty much the entire tubule itself, the entire tubule is about only 1.2 inch, sometimes an inch long. The entire tubule consists of a single layer of epithelial cell, some of it being cuboidal and some of it being squamous. This part here, I should know, is actually squamous because they are thinner. The rest of them are cuboidal. You could see some of them here getting into the collecting ducts. Pretty much, instead of becoming cuboidal, they could become a little bit longer, but not necessarily columnar, but just a little bit longer. So you have several tubules, several parts. The tubule itself has three major parts. You have the proximal convoluted tubule. There is your proximal convoluted tubule, okay? And after the proximal convoluted tubule, well, the proximal convoluted tubule is called proximal because it's the closest um, to the capsule itself. Then you have the nephron loop. This was actually called the loop of Henle. Okay, so after the nephron loop, you have the distal convoluted tubule right there. That's your distal convoluted tubule, which is actually now going to enter or be connected to the collecting, actually your collecting duct. The collecting duct will empty the urine that it has filtered right into the minor calycine. Okay, it is called distal convoluted tubule because it's the farthest from the renal capsule, and it drains into the collecting duct. Let's take a look at the next one. Let's take, so the nephron loop, right? This is actually the loop itself. The nephron loop is pretty much like a loop, formerly called the loop of Henle. When I was studying, we call it the loop of Henle. It's a U-shaped structure consisting of two limbs. Excuse me. One limb, right, this one here, is actually immediately connected to the proximal convoluted tubule, is the descending limb. And it's made up of the descending thin limb, and it's lined with simple squamous epithelium. And the ascending limb. The ascending limb is, there you go, that's the ascending limb. Okay, from the thin limb, it goes essentially into uh, becoming lined with cuboidal or columnar cells, like in this area here. Okay, and finally, your distal convoluted tubule, which is also lined with cuboidal cells with very few microvilli. They function more in secretion rather than absorption. I'm going to discuss with you what is meant by the word secretion in kidney or in urine formation and what is meant by um, filtration and then reabsorption in kidney function. Okay. Now, in the collecting ducts right here, it is just um, worth to mention some very important cells. One of the important cells right here is what you call the principal cell. That's your principal cell. These are the cells of your collecting duct. Now, the principal cells are sparse with short microvilli, and they are responsible for maintaining water and sodium balance. Then you have also the intercalated cells. Intercalated cells are of two types, A and B. Both of them help maintain acid-base balance in the blood. The collecting ducts receive filtrate from many nephrons, and therefore this is not only the one nephron that would serve. You have other nephrons that would pretty much be collected by the collecting duct itself, which will eventually be running right through the medullary pyramids, which gives the pyramids their striped appearance. It's actually the collecting ducts, large as they are, will be the ones that's giving the pyramids the striped appearance. The ducts fuse together to deliver urine through the papilla, which are actually opening the drain itself into the minor calyces. Now, this is a very good histological picture of the Bowman's capsule, the glomerulus. This is the tuft of capillary, which you call the glomerulus, and they're all actually made up of lined with simple squamous epithelium. But the beauty is that you can see the parietal um, area of the Bowman's capsule that's lined with simple squamous. Can you see that? That line right there? That's actually the simple squamous. They're flat, okay, and they are actually lining they're lining the Bowman's capsule itself. The Bowman's capsule empties into mm, I'm looking at where it's actually emptying. Is it here? Okay. Well, I'm looking at this one because this is where your proximal convoluted tubule is. So I'm looking at this is actually where it's actually emptying. No. Oh yeah, this is where it's emptying. You have the distal convoluted tubule right there. Okay, because the distal convoluted tubule pretty much like connects with a unit right here, which you call the juxtamedullary apparatus. This is your juxtamedullary apparatus, which I will be discussing with you later. So majority of the kidney cells lining the tubules are actually made up of simple cuboidal epithelium, a single layer of cuboidal cells, except for the glomerulus, which is simple squamous, except for the wrappings of the outside layer of the capsule itself, which is simple squamous. And then let's move on. Then I can show you now, pretty much, we can actually progress into looking further at the entire kidney itself. Do we have only one type of nephron, or are there different classifications? Is there a classification of neurons, or are they the same? Well, to me, they all look like the same, but because of one particular difference in function, as well as in not only physiology, but as well as in anatomy, there are really two different types of nephrons. These are your cortical nephrons. This is your cortical nephrons, this little tiny guy there. And it makes up about 85% of the nephrons in the kidney. And they are located mostly in the cortex with very little, loop of Henle sticking out. So your that's why that's how they got the name cortical nephrons. Then you have the juxtamedullary nephron. This is your juxtamedullary nephron. They have long loop of Henle or nephron loops. You see how long they are, and they invade the medulla, almost the entire medulla, all the way to the bottom. Okay, and the ascending limb. Okay, the ascending limb. Let me see. This is your ascending limb. Whoops, okay? The ascending limb have thick and thin segments, and this nephron type is important in the production of concentrated urine. It is just really obvious. This is how you concentrate right through the loop of Henle, is how you concentrate the urine. A lot of water is absorbed right through that particular area, and a lot of sodium is actually being reabsorbed, secreted and reabsorbed. There's actually secretion and reabsorption, selective secretion and reabsorption of both water and sodium in that area. So if you have reabsorption of water along a longer route, which is the route for the loop that you find in juxtamedullary nephron. This particular type of nephron is actually the one responsible for forming concentrated urine. This cortical nephron is responsible for forming diluted urine. Okay, so the renal tubules are associated with two capillary beds, and the capillary beds will be your glomerulus. You know what happens there. The filtration of the urine happens there, or the blood rather, happens there to form the filtrate that becomes the urine. Then you have the peritubular capillaries. These capillaries here, you could see capillaries, and these capillaries here also, the peritubular capillaries, are the ones that are associated with the tubules in order to assist in the reabsorption and absorption. Remember that in the filtration system itself, you are filtering the entire blood itself. Therefore, the efferent, what you call is the efferent blood, that's going to be picked up right here in the after the glomerulus, it's going to be really one thick blood. You lost a lot of your water, but you wouldn't want to pretty much like lose that water to your urine. You're not really losing all of your water through the urine. Your water, once it gets filtered, your blood, the water part has been filtered off, will be reassessed by the tubules and will be reabsorbed back. Now, during the face-to-face, I usually give an analogy as to exactly how my students will understand the function of the filtration mechanism or the formation of urine in the kidney. Well, I give the analogy of the husband and wife who is actually like arguing because they have so many clutter, so many clutter and so many garbage in their garage. And so they said we should clean our garage so we can actually have a better looking garage and our cars can actually get into it, because the clutter that we have accumulated through the years is actually, there may be a few things here that we can already throw away. So what they did was, okay, let's try to empty our garage. So they emptied the garage, which is actually like what our filtration mechanism is. Your entire blood is going to be filtered by the nephrons. About 1,200 ml per minute of it is being filtered by the nephrons. So this husband and wife emptied their garage, and then they decided, hey, we cannot just throw away everything. There should be some things here that is actually very vital to us. So slowly they tried to pick in some of the stuff back into the garage, because they're saying a lot of them have memories in them, and therefore they don't want to throw them away. Okay, so and some of them are just basically something that they use only once a year, and they have to keep it right there in the garage. So they practically like pretty much like eh, put in right back half of what they're pretty much like emptied out. So they said, okay, so this particular half here that we emptied out, are we going to empty this out? No, we're practically going to probably take a look at them, sift through them, and some of them are really, really pretty old. Some of the things that they got back here are pretty old, and therefore they need to pretty much like throw them away, regardless of whether they have memories. They're pretty old, so they are something that is only like dust collectors, and therefore they have to throw them away. So they now analyze the importance of everything that they need to take in. Some of them they take in, then they move it back again outside. They decide that it's not really worth keeping them for another 10 years or another year. So that's exactly how the selection mechanism goes also in the reabsorption of filtrate, the solutes in the filtrate, and then the secretion, meaning the throwing away finally. So that happens between the loop of the entire length of the tubule, right? So you have that's why you will need blood vessels. You need blood vessels to pretty much like take in the ones that gets reabsorbed back. And right through the blood, they might have just reabsorbed something that they need to really throw away. So it needs to be like that blood needs to be in contact again with the nephron tubes to push back again what they need to really throw away. So these capillaries, which is your peritubular capillaries right here in the cortical nephron and the vasa recta, are low pressure porous capillaries that is adapted for absorption of water and solutes. They arise from the efferent arteriole. So you have your efferent arteriole right there. Then the efferent arteriole, you're assuming, is actually going to be uh, like delivering blood here in this section here that is really very concentrated because you just lost a lot of the water into the filtrate. So these capillaries cling to the renal tubules in the cortex and empty into the venules. The vasa recta, this is actually the vasa recta, these are also thin-walled vessels that are parallel to the long nephron loops of the juxtamedullary nephrons. They arise from the efferent arterioles as well. Okay, they arise from the efferent arterioles, and they serve the juxtamedullary nephron instead of the peritubular capillaries. The vasa recta functions in the formation of concentrated urine. Let me repeat that. Make sure that you know. And the diluted urine is a function. Formation of diluted urine is a function of the cortical nephron. And about 85% of our nephrons are cortical nephrons. You could just imagine. So we pretty much like produce urine that is not really very concentrated. So the glomerulus, let's now focus into the glomerulus. These are capillaries. This is your efferent arteriole and the afferent arteriole. Blood goes from the afferent, okay, through inside and into the glomerulus area where filtration is going to happen, and the efferent blood exit through the efferent arteriole. This is where they're going to join in either the peritubular capillaries and also into the vasa recta if it is actually a juxtamedullary nephron. So these glomerulus are specialized for filtration. They are different from the capillary beds because they are fed and drained by the arterial. The capillary beds on the skin is actually fed into the blood feeds into the capillary by the arterioles, and then the venules pick them up. Here, no. It is actually an arteriole that feeds blood into the glomerulus, And then another set of arterial, which is the efferent, is actually going to pick up the blood. The afferent arterial enters the glomerulus. That is your afferent arterial. The afferent arterial arises from the cortical radiate arteries. And the cortical radiate arteries actually is a branch of the uh, is actually a branch of the arcuate arteries. So from the arcuate arteries into the cortical radiate arteries into the afferent arterioles, and pretty much they empty into the afferent arteriole now empties blood um, into the glomerulus. The efferent, on the other hand, feed into the peritubular capillaries. This one feeds blood into the peritubular capillaries, or the vasa recta if it is a juxtamedullary nephron. Blood pressure inside the glomerulus is high, simply because if you're going to concentrate on looking at these two arteries, the afferent and the efferent, blood enters through the afferent, and you have a wide afferent arterial. Can you see how wide the afferent arterial is? Comparing it with the efferent arterial. The efferent arterial is narrower. If you have an exit point that is narrower and your entry point is wide, imagine your cars, okay, getting into a highway, uh, coming from a big highway, it goes into a smaller highway, and the exit point of that highway is only one, and it's actually like narrower. What's going to happen within these particular tributaries or highways? Well, there's going to be accumulation of blood. The pressure inside it is going to be increasing. So because of that increased glomerular pressure, then some of the blood now, the water portion of the blood is now going to be pushed outside. And now we will be discussing how that particular filtration is actually happening when we get into that point. So the afferent arterioles are naturally larger in diameter than the efferent arteriole. And the arterioles are high resistance vessels. So each nephron has one juxtaglomerular complex. Ooh, that's another name. That's a new name. This is your juxtaglomerular complex right here. The juxtaglomerular complex is made up of macula densa cells. That's your macula densa cells, which you will need to identify also histologically, okay? And these are the macula densa cells of the ascending limb of nephron. So you have your ascending limb of the nephron, which is actually part of the what do you call this, the loop of Henle, the ascending limb of the loop of Henle. They are actually butting in and joining in that particular, it's like a trio between the afferent arteriole, the efferent arteriole, and then the ascending loop of Henle. And then as they connect, they realize that there's actually specialized cells there that they see that seem to really talk to each other. Okay, so you have the macula densa cells. This is part of the ascending limb of the loop of Henle. You have also the extra glomerular mesangial cells or mesangial cells. That is actually part of the afferent arteriole itself. Then you have the granular cells. That is actually right here. You're probably wondering, how would the kidney, does the kidney have the brain? How would the kidney know how much urine needs to be produced? Would the kidneys be producing urine all the time, even if our blood pressure is very low? Let's say you get into an accident, you hurt yourself, you're losing blood, and your blood pressure is so low. Are you going to be producing urine still? That's gonna kill you. I mean, you're already losing blood, and then yet you're producing urine. Man, how much water in the blood is going to be left? What if you're dehydrated? You just had really a bad case of diarrhea. Having a bad case of diarrhea, that's really more common than having an accident. You know that if you have a bad case of diarrhea, your urine formation stops or it slows down. And who does that? Does the kidney have a brain? Well, this unit here, the juxtaglomerular complex, I'd say, in my opinion, okay, is the brain of the kidney, or is one of the brains of the kidney that helps in deciding how much urine needs to be produced, okay, by the kidney, and when it's actually going to be speeding up or slowing down. So the juxtaglomerular complex involves modified portions, and therefore the cells there, like the macula densa and the mesangial cells, are actually like modified. So they are part of the distal portion of the ascending limb of the nephron, and they are abutting the afferent, okay, and sometimes they're also abutting the efferent, and they are important in regulating the rate of filtrate formation and blood pressure. And the population of the cells there will be your macula densa. That's very important for you to know, and even in your histology. These are tall, closely packed cells of the ascending limb. You could see how different they are. Tall, closely packed cells of the ascending limb. They contain chemoreceptors. The macula densa contain chemoreceptors that can sense sodium chloride content of the filtrate. Then you have your granular cells. The granular cells are actually the ones that are lining the afferent arteriole. They are enlarged and smooth muscle cells of the arteriole. They act as mechanoreceptors. They could sense the pressure of the incoming blood that goes right through the afferent arteriole. So they sense the blood pressure. And then they also contain secretory granules that release the enzyme renin, okay, when needed. Now, the extraglomerular cells or the mesangial cells, I'm having a hard time. I'm having a hard time pronouncing the extraglomerular cells. I call it that mesangial cells. So mesangial cells, this one here, right there in the center, okay, pretty much is located between the arterioles and the tubules. The arterioles and the tubules, that one here. And it is claimed to be pretty much the link, the information link between the macula densa and the mesangial cells. They are because it is interconnected to these areas with gap junctions. And they are the ones passing signals between the macula densa and the granule cells. So how efficient are these? Well, they are very efficient. About 180 liters of fluid. My goodness, how much is 180 liters of fluid? Is processed daily. But actually, 100 out of that 180 liters of fluid that goes through our kidneys daily, only 1.5 liters of fluid per day is formed. You could just imagine what kind of selection in the reabsorption process. The filtration is just mechanical filtration. Because of the pressure, they all actually leave the blood and they get into the tubules. It is the manner by which these tubules would selectively reabsorb those that needs to be reabsorbed, so that you only have 1.5 liters that you need to excrete per day. So the kidneys filter the body's entire plasma volume 60 times each day. And the kidneys consume about 20 to 25% of oxygen that is used by the body at rest. The filtrate that is actually the one produced by glomerular filtration is basically blood plasma minus the proteins. Proteins cannot get into the urine, all right? They're bigger. So if you see proteins in the urine, that is not good because you're not supposed to filter urines. There's actually times when you can see that, most especially when you just exercise, but it's not protein, some amino acids and very, very tiny proteins. You can actually find it in the urine, most especially even after an exercise. Urine is produced from the filtrate that's actually running right through the tubules. So urine is 1% only, even less than 1% of the original filtrate that was filtered through by the glomerulus. And the urine contains metabolic waste and substances that we don't need. And if they're water soluble and they're not needed and there's too much, then they go out. All right. These now are the three major renal processes that is happening in the glomerulus. You now have a short version of your nephron. You have the capsule, the renal capsule, and then you have the tubes. All right. This is now going to be a representative of the entire tube itself. You have your proximal convoluted tubule, the loop of Henle, and the collecting tubule. But at the most proximal end is actually your nephron itself. So what goes on in the nephron? Three processes are involved in urine formation and adjustment of blood composition. First is actually glomerular filtration, and the glomerular filtration happens right here in this section, which is actually where you find the glomerulus. It produces cell-free and protein-free filters. So this area here, this filtering mechanism right there in the glomerulus is very important that you know that they're so efficient that they will not allow cells and proteins to pass through and be filtered through. Then you have also what you call tubular reabsorption. This is the selective return, about 99% of the substances that is part of what was filtered through the glomerulus. 99% goes back to the blood. That's why this efferent arteriole is now connected to the peritubular capillary. The peritubular capillary receives those substances that is reabsorbed back into the bloodstream. And it's actually returned. About 99% of them is returned back into the blood via from the renal tubules into the peritubular capillaries or the vasa recta. Then you have the third process, which you call tubular secretion. So the green line right there is tubular secretion. So this green line here is pointing to tubular secretion, where substances that you have already actually returned to the blood now is going to go back to the tubes and become part of the urine. Tubular secretion is the selective movement of substances from the blood back to the filtrate in the renal tubules and collecting ducts. And pretty much this entire process is here is what you call the major renal processes. At the top of it is glomerular filtration. This section here where you have the glomerulus is a passive process. There's no magic in there. No metabolic energy, no ATP that's actually required. The only thing that's actually happening there is you have hydrostatic pressure of blood because of the um, wide afferent arteriole that's picking up a lot of blood, and then the narrow efferent arteriole that's actually preventing blood from escaping. So you have an increase in blood pressure right here. And because of the nature of the glomerulus being fenestrated, then it will allow really water and filtrate to pass through it, except for cells and proteins. So the hydrostatic pressure there would force the fluids and solutes through the filtration membrane into the glomerular capsule. That's actually your capsule. That's the number one right there. There is no reabsorption into the capillaries that occurs in the glomerulus. And so the porous membrane between the blood and the interior of the glomerulus capsules allow water and solutes smaller than the plasma proteins to pass, and normally no cells can really pass. The filtration membrane, this filtration membrane here, I'm sorry, the filtration membrane contains three layers. First is actually your fenestrated endothelium of glomerular capillaries. You have the basement membrane, which is the fused basal laminae of two other layers, and what it calls um, an interior or a second membrane that covers and butts in and is actually hugging the glomerulus itself, which you call the layer of the visceral layer, okay, or it's actually made up of specialized cells which you call podocytes. And these podocytes form filtration slits, and they do repel macromolecules. Let me take a look at some pictures, so maybe we can have podocytes somewhere here. There you go. This is your glomerulus, and pretty much this picture here shows you the filtration membrane. Now, glomerular filtration, which happens around this area, is actually controlled by your afferent arteriole and your efferent arteriole, the pressure inside them. But what actually pretty much selects the substances that gets filtered through and becomes part of the filtrate is actually the filtration membrane. The filtration membrane is made up, first of all, by the glomerular walls of the glomerulus itself, which is simple squamous epithelium, and it has fenestrations and quite a little bit selective, but more selectivity is actually happening um, because the visceral covering that pretty much covers the glomerulus is actually made up of specialized cells which you call podocytes. So the filtration membrane, there's no reabsorption into the capillary, but the filtration membrane pretty much is made up of porous membrane uh, between the blood and the interior of the glomerulus capsule and allows water and solutes smaller than plasma to pass through. And let me give you a little bit more of that filtration membrane. There you go. This is one short segment of the glomerulus. You have your glomerular capillary, which is a fenestrated capillary. It has a lot of holes in it. And that lot of holes really is going to pretty much like uh, allow a lot of substances to leak right through it. Maybe protein will leak through it. But protein is being prevented from leaking through because this particular specialized visceral cover, which is made up of podocytes. You could see a podocyte having a lot of um, legs, okay? And they form, the food processes form a sieve or another filtering layer that filters large protein off. And this is actually the filtration slits of the podocytes. So you have three layers of filtrating mechanism. You have the fenestrated endothelium of the glomerulus. You have the basement membrane which is actually the fused basal laminae of two other layers. Then you have the food processes of the podocytes with filtration slits. Pretty much, this would repel macromolecules such as protein. And this is by electron microscopy. This is a closer look at the membrane under electron microscope, and it shows the food processes of the podocytes with filtration slits. And the slit repels big molecules. It only allows molecules less than 3 nanometers to pass through, such as water, glucose, amino acids, and some nitrogenous waste would be allowed. Okay, uh, would not be allowed, I think. These are big. Glucose should not be allowed. And the amino acids, but then there are some really like small amino acids that could be, okay? Plasma proteins remain in the blood to maintain the colloid osmotic pressure. You cannot really like release plasma protein and excrete it. So if you lose your protein, plasma protein, you're going to lose also the water uh, of your plasma. You want to maintain the colloid osmotic pressure of the blood to prevent the loss of water into the capsular space. Okay? Proteins in the filtrate, if they get released to the urine, indicates a problem. Now, there are some macromolecules that get stuck right there into the filtration membrane, and pretty much you don't have anybody to clean it up but your uh, glomerular mesangial cells. The glomerular mesangial cells are white blood cells, they're macrophages, and they pretty much clean up that particular area. So now let's take a look at the filtration that's happening along the glomerulus and let's try to understand how the pressure of the body affects the pressure or the filtration in the kidneys. Okay, well, again, let me tell you, uh, no matter how it is, whatever pressure is actually filtering the blood right through the glomerulus, there's no magic in it. It's just hydrostatic pressure that's pushing blood fluid right through. Okay, so you have your net filtration pressure. The net filtration pressure, the pressure is actually the one that would pretty much like produce the filtrate that goes right through the tubule. Okay, and the net filtration pressure pretty much is um, affected by several parameters. First, you have your outward pressure. The outward pressure, okay, the outward pressure is the pressure that pushes the water to go out. And then you have also the inward pressures. The inward pressures is actually the pressure that would prevent the water from going out. Uh, what is it? Well, first of all, let's take a look at the outward pressure. The outward pressure pretty much is your force that promotes the formation of the filtrate. So whatever force you have here, it forces the water to go in. The hydrostatic pressure itself, we call it hydrostatic pressure in the glomerular capillaries. Pretty much it is essentially the blood pressure and the chief force that's pushing the water and solutes out of the blood right through the glomerulus. And it is really quite high. Okay, regardless, it's actually an average of about 55 millimeters of mercury pressure. That's compared to pretty much like about 26 millimeters of mercury pressure in the capillaries. Our capillary have 25 millimeters of mercury pressure in it. That helps in the exchange of food between the capillaries and the blood and the tissue, exchange of materials. In the kidneys, in the glomerulus itself, the uh, the capillary force is about 55 millimeters of mercury pressure. That and that is present right here in right, right here actually in the afferent arteriole. Now, the reason for this 55 millimeters of mercury pressure is that the efferent arteriole is a high resistant vessel with a diameter that is smaller than the afferent arteriole. So the hydrostatic pressure is actually created by the amount of blood that is actually pumped in with a pressure of 55 millimeters of mercury, and it goes right through a narrower arterial, and therefore there's going to be a buildup of pressure right here. Okay, so that buildup is is actually the glomerular pressure, which is now going to be 55 millimeters of mercury pressure. That's the one pushing the filtrate to go out. That's the hydrostatic pressure in the uh, in the capsular space. Do we have hydrostatic pressure in the capsular space? Of course. This capsular space is not empty. It has already a pre-existing fluid inside it. So when you're pushing fluid from the glomerulus, from the coming from the afferent arteriole, and there's a buildup of pressure here, then that particular movement of water crossing the glomerular capillaries will be met by an opposing force that is already present here, which is the hydrostatic pressure of the capsular space. Okay, so you have your hydrostatic pressure in the glomerulus, and then you have the hydrostatic pressure in the capsular space. That is actually preventing that from moving. It prevents the filtrate from going in. Okay, which is about 15 millimeters of mercury pressure only. So you're going to deduct that 55 millimeters of mercury pressure. You're going to deduct this 15 millimeters of mercury pressure, deduct it from 55. That's going to be the net, right? 35, right? But then you have also another opposing pressure, which is the colloid osmotic pressure. The colloid osmotic pressure in the capillaries, which is actually the osmotic pressure uh, in the colloid, will pull the proteins in the blood. This colloid is holding on to the items that is actually already in the plasma, and they don't want them to escape. Therefore, it says some of you can go, but some of you must stay here. These are your colloid osmotic pressure. It is actually preventing the loss of water and so many things from the blood. Okay, which is about really, it is really a very strong pull, about 30 millimeters of mercury. So these two, where their arrows are all going to be resisting the hydrostatic pressure that moves water into the tubules, these two forces here are actually the inward forces, okay, that would prevent the formation of the filtrate. So the net filtration pressure, therefore, is the resulting force responsible for filtrate formation and is the main controllable factor determining the glomerular filtration rate. Mathematically, the net filtration pressure is actually expressed as the outward pressure minus the inward pressure, the total inward pressure. And the outward pressure is actually your hydrostatic pressure in the glomerulus. That's the push of the water and the pressure in the glomerulus going out, and is met by forces that would not allow it to go further, okay, which is your colloid hydrostatic pressure, okay, and the pressure of the capsular space, the pre-existing pressure in the capillary space, which is the pre-existing hydrostatic pressure already. Together, okay, the HP plus the OP will be your factor that opposes the what do you call this, the formation of filtrate. So you add them together, you have only a balance of 55 minus, well, 15 plus 30 is 45. 55 minus 45 is 10 millimeters of mercury pressure. So that is only 10 millimeters of mercury pressure is the only balance net filtration pressure, the balance of the pressure that is going to make you produce your urine. All right. So if your blood pressure drops, if your blood pressure is about 100, 120 millimeters of mercury pressure, and your kidney is used to that, and then all of a sudden your blood pressure drops, instead of 120, your blood pressure becomes 90. So that's a good drop already. Eventually, this what do you call this hydrostatic pressure is also going to be diminished. When the hydrostatic pressure is diminished because of um, a decrease in your total blood pressure, then you may not be producing urine at all, or the ability of your kidneys to produce urine by way of filtration is not anywhere going to be effective. So your urine production is minimized, okay, if your blood pressure drops because of whatever reason. So right here is actually like a good picture of how the glomerular filtration rate, or the what we call GFR, is actually regulated. And pretty much, I would like you to take a look at this and study very well what happens. And this is only when the systemic blood pressure goes down. When the systemic blood pressure goes down, for whatever reason it goes down, usually there is a homeostatic response by the body, so that we will not lose our ability, or the kidney's ability, to produce urine. For instance, uh, let me just go and pretty much like, well, see, if I discuss this with you now, you are not anymore, well, you will not be able to understand it, because um, you may not know yet exactly what these things are. Okay, but I will be looking at that. Okay, let me see. Let me look at this. Okay, when your systemic blood pressure drops, your glomerular filtration rate is also going to drop. And that means the filtrate flow and the sodium chloride flow in the ascending limb of the nephron is also going to drop. Okay, now this particular reaction would act on the macula densa of the juxtaglomerular complex of the kidney, which eventually will pretty much like stimulate the release of vasoactive chemicals, or release of vasoactive chemicals, uh, which will result in the release of vasoactive chemicals, uh, which will result in the inhibition of the release of vasoactive chemical. Okay, I said it right. Which eventually is going to result in the release of in the inhibition of the release of vasoactive chemicals. And pretty much the result is actually vasodilation of the afferent arteriole, which would increase the GFR. Or if your systemic blood pressure drops, okay, the granular cells of the juxtaglomerular complex of the kidney will feel it. Okay, this will stimulate the release of renin, and renin pretty much like would catalyze the formation of angiotensin 2 from angiotensin 1. Okay, and then uh, pretty much from angiotensin 2, once angiotensin 2 is around, aldosterone is stimulated, and that results in the vasoconstriction of the systemic arterioles, and that will increase peripheral resistance. If peripheral resistance is increased, remember your blood blood vessels, guys. Okay, when peripheral resistance of the blood vessel of the blood is increased, blood pressure is increased. So blood pressure is increased, and that's the effect of angiotensin. And uh, one effect of angiotensin is increased secretion of aldosterone that's coming from the adrenal cortex. And this is a different organ that's actually sitting on top of the kidney. And the result will be increased sodium reabsorption by the kidney tubules and water will follow it. Okay, and then your blood volume will increase. And once blood volume is increased because of aldosterone, then systemic blood pressure will increase, which is actually a homeostatic mechanism that would reverse the decrease in blood pressure. So the decrease in blood pressure, when something happens to you drastically, is not really going to completely stop. It will slow down your information, but eventually all of these reactions will kick in that will pretty much like resume our kidney's ability to form urine. So please make sure that you understand and you take a look at those things. So the glomerular filtration rate, this is actually an overview of how the GFR is actually regulated. The GFR, or glomerular filtration rate, happens to be the volume of filtrate that is formed per minute by both kidneys. And a normal volume is about 120 to 125 ml per kidney. Wow, that is a lot. The GFR is directly proportional to another acronym, NFP, or net filtration pressure. So the net filtration pressure is actually where the primary pressure it's actually the primary pressure um, that is happening which is actually in the glomerular hydrostatic pressure. And I just gave you the mathematical expression of the net filtration pressure the um, uh, in the slide before this. So the total surface area for filtration is controlled by the contraction of the glomerular mesangial cells. The filtration membrane permeability is much more permeable than other capillaries, and the control of the GFR is divided into two sections. First, you have your intrinsic control, which is also known, your intrinsic control, which is also known the intrarenal, okay, or renal autoregulation. And you have your extrinsic control. This section here is the extrinsic control or extrinsic mechanism, which is the hormonal and neural mechanisms. The intrinsic controls function locally within the kidneys. This part here functions locally within the kidneys to maintain a relatively constant glomerular filtration rate despite the normal fluctuations in systemic blood pressure. These are the ones that make sure that your kidneys will be doing their job despite the fluctuations around. The extrinsic control, however, are systemic responses that primarily maintain the overall blood pressure and can override the intrinsic regulation in extreme circumstances, such as significant blood loss, etc. Okay, so the goal of your intrinsic control is to maintain the GFR in the kidneys. The GFR affects systemic blood pressure. An increase in GFR causes increased urine output, which lowers blood pressure, and vice versa. Now, the goal of the extrinsic controls is to help in maintaining systemic blood pressure. The nervous system and the endocrine mechanisms are the main extrinsic uh, controlling mechanism for the extrinsic control. Okay, now let's take a look at the intrinsic. Okay, now we're going to get into the GFR regulation. We start with the intrinsic, then we get into the extrinsic. The extrinsic controls of renal regulation pretty much maintains nearly a constant GFR when the mean arterial pressure is within the range of 80 to 180 millimeters of mercury. So only within that part. If it is too low, it's lower than 80 or higher than 180, your intrinsic control is not going to happen in such extreme cases. The auto-regulation of the intrinsic mechanism would cease if they're out of range. So there are two types of auto-regulation, renal auto-regulation. One is what you call myogenic mechanism, and the other one is what you call the tubulo feedback mechanism. I will be discussing them one by one. This is just a very good view of both the myogenic, this is a myogenic mechanism. It relies on the inherent properties of the arterial, and you have the tubulo-glomerular feedback mechanism that relies on the interaction between the nephron tubule and the glomerulus itself. Now, myogenic relies on the properties of the arterial. Okay, let's take a look at that. You will see that again. So let's take a look at the myogenic. First one is the myogenic. The intrinsic control. Myogenic is intrinsic control of renal autoregulation. The myogenic mechanism is an inherent property of the smooth muscles of the afferent arterial. So it's actually the smooth muscles of the afferent arterioles. They respond to high blood pressure. Increased blood pressure stretches the afferent arteriole's smooth muscle, causing it to contract. And when they contract, they undergo vasoconstriction. This vasoconstriction reduces the blood flow into the glomerulus, and thus it will eventually decrease the glomerular filtration rate. So these smooth muscles here can regulate what's going to be filtered off by just the contraction of the smooth muscles. That is in response to high blood pressure. Now, in response to low blood pressure, when the pressure drops by any reason, the arteriole will relax and would dilate, increasing blood flow into the glomerulus to bring the glomerular filtration rate back in. The next one is what you call the tubulo-glomerular feedback. This mechanism involves the juxtaglomerular apparatus, which means your macula densa, your mesangial cells also, and the granular cells, where the distal convoluted tubule and the afferent arteriole really meets. You have your distal convoluted tubule, okay, and the afferent arteriole would meet, and they form a triad, which is the juxtaglomerular apparatus. Now, in response to high juxtaglomerular apparatus, when the GFR is too high, the rapid flow of filtrate results in less absorption of sodium chloride in the proximal tubule. Specialized cells in the JGA or juxtaglomerular apparatus, which you call the macula densa, would sense this is actually your macula densa, it would sense this elevated sodium chloride concentration. The macula densa then releases local vasoconstriction like ATP and adenosine that constricts the afferent arteriole, which eventually will decrease the GFR. Now, what if it is the other way around? What if there is actually low GFR? So when GFR is too low, the flow of filtrate is slow, and most sodium chloride is reabsorbed. The macula densa detects how low sodium chloride concentration and how low the sodium chloride concentration is and signals for vasodilation of the afferent arteriole, which will now increase the GFR. So your tubulo-glomerular feedback mechanism is actually a flow-dependent mechanism that directs the macula densa, therefore, to respond to the filtrate sodium chloride concentration. Then again, let me repeat it again. That's why I put it here. It's a different explanation of just what I said. If the GFR increases, the filtrate flow rate increases. It leads to decreased reabsorption time, causing the sodium chloride level in the filtrate, high sodium chloride level in the filtrate. So the feedback mechanism causes constriction of the afferent arteriole, which lowers the net filtration pressure and GFR, allowing more time for sodium chloride reabsorption, which is actually opposite mechanism that's happening if there's going to be a decreased GFR. So now we're going into the extrinsic control of the GFR. Guys, I know that this becomes overwhelming because you will need to pretty much like go through this mechanism, allow yourself to absorb what is really happening, allow yourself to understand what this GFR and net filtration pressure is, what this tubular thing is, and etc. You have to allow yourself to memorize the parts of the nephron, what the macula densa mesangial cells are, so that you will understand it. So please make sure that you do that, because just hearing it from me and you're not going to be reviewing the parts, you will not understand it. So let's take a look at the extrinsic control of the glomerular filtration rate. So this is the external regulation involving the nervous and the endocrine system, and is triggered by changes in the overall blood pressure, and this one overrides the intrinsic control. When it is the intrinsic control is automatic, the automatic renal. Whenever it is way beyond 80 to 180, and automatically that automation is not going to happen anymore, what actually happens is that the extrinsic control will take over. And it is controlled by the sympathetic nervous system. For instance, under condition of stress, such as severe blood loss or exercise, the sympathetic nervous system is activated. The effect will be a strong sympathetic stimulation that causes the release of your sympathetic hormones, epinephrine and norepinephrine, which induce really a potent vasoconstriction of the afferent arteriole. So that's your afferent arteriole right here. Okay, they are already narrow, but they become narrower. These would dramatically reduce the flow of blood into the kidney, shunting it to more vital organs like the heart and the brain, and causes a very steep drop in the glomerular filtration rate. So the purpose of the extrinsic control is to regulate the glomerular filtration rate to maintain the systemic blood pressure. If there's something wrong with the systemic blood pressure and it's affecting the way your autoregulation is actually filtering, and the forces there, the blood pressures there are actually like not within range, then your extrinsic mechanism would have to maintain systemic blood pressure through the neural and hormonal mechanism. Extrinsic controls will override the renal intrinsic control if the blood volume needs to be increased. Okay, the sympathetic nervous system under normal conditions at rest, okay, the renal blood vessels will be dilated with sympathetic nervous system, and the renal autoregulation mechanism will actually prevail. So pretty much you will have to make sure that you remember all of this and this pathway, guys, so that you will understand the extrinsic control. Then now will be the control of the hormones. Instead of just the sympathetic nervous system, now will be your hormones. Your renin-angiotensin regulation is hormonal. Renin is a hormone produced by uh, the cells of the kidneys, and this is actually responsible for maintaining also uh, um, um, maintaining also the GFR. So this is the key hormonal mechanism for long-term blood pressure regulation. Activation of these RRAs, renin, angiotensin, aldosterone system, happens when blood pressure drops. So when the blood pressure drops dramatically, the juxtaglomerular cells in the kidneys would release the enzyme renin. So it is the JGA cells that will release renin. Renin would initiate a cascade that produces angiotensin 2, which is a powerful vasoconstrictor. So, and with the help of renin, angiotensin, okay, angiotensin is produced by the liver. Angiotensinogen, which is the inactive form, is produced by the liver. Renin acts on angiotensinogen to form angiotensin 1. And then your ACE, which is actually your angiotensin converting enzyme, will convert angiotensin 1 into angiotensin 2. The angiotensin converting enzyme is actually an enzyme that's produced in the cells of the lungs. So now you have angiotensin 2. The angiotensin 2 will now vasoconstrict the arterioles. Okay, and that's going to happen. And you have a lot of angiotensin 2 receptors in the efferent arteriole that's going to be receiving and reacting to the presence of angiotensin 2. This will pretty much like constrict it. Well, actually, it's also going to be constricting also the afferent, both the afferent and the efferent arterioles. But it will be constricting the efferent arteriole more forcefully than the afferent, which will help to maintain or even increase the glomerular filtration despite the low systemic pressure. So you do not really lose the ability, your kidneys do not really lose the ability to form urine immediately after you you got into what they call it something happened to your body and your blood pressure drops. You're still going to produce it because this the renin angiotensin system will kick in, and this will kick in when your blood pressure drops. Okay, so it stimulates aldosterone through the angiotensinogen. Um, it stimulates the release of aldosterone, which increase sodium and water reabsorption. And when, remember this, guys, where there is water, or where there is salt, rather, where there is salt, water follows. Okay, so when sodium is reabsorbed, water will follow through. And thus, raising the blood pressure. So this happens whenever the renin-angiotensin system will happen whenever blood pressure drops. Then you have another hormone which you call the atrial natriuretic peptide. It's a hormone that works to lower blood pressure. Most of the hormones that we're going to uh, look at that would affect blood pressure tends to increase blood pressure, except for the atrial natriuretic peptide. It's a hormone that works to lower blood pressure and increase your urine formation through the increase in GFR. It, it is actually released by the atrium of the heart in response to high blood volume and pressure. And the effect of this peptide, it is produced by the atrium of the heart, will be to promote vasodilation of the afferent arteriole. Yeah. The afferent arteriole is going to dilate, and inhibits also the release of renin and aldosterone, because so that this eventually this leads to the increased glomerular filtration rate and urine production, which lowers blood volume and systemic blood pressure also. Make sure that you know the atrial natriuretic peptide. It's already here in your picture here. It dilates the afferent arteriole, leading to increased blood flow and filtration, which is eventually going to lead into diuresis. Okay. The AMP suppresses reabsorption of sodium in the collecting duct and other segments of the nephron, which is actually going to result in natriuresis. There are also other factors affecting GFR besides the factors I mentioned to you. The renal cells release a variety of chemicals. Some chemicals act as paracrines that affect renal arterioles, such as adenosine, prostaglandin E2. Some cells make their own locally acting angiotensin II, which reinforces the effect of hormonal angiotensin II. These are all part and parcel of the extrinsic pathway. And here you have a summary of the regulation of GFR in a table, which helps you in your review. Please make sure that you go over this. Okay, and there is one last picture here from a last slide, uh, an abnormality which we call anuria. It's an abnormally low urinary output, which is less than 50 ml per day. It may indicate that the glomerular blood pressure is too low to cause filtration. And renal failure and anuria can also result from situations in which the nephrons would actually stop functioning. An example of this is acute nephritis and transfusion reactions. And of course, crush injuries. And I mentioned to you that your, most especially your right kidneys, is actually very susceptible to physical damage because it's not really fully protected. All right, and I think that should be it. That should be it. And I will see you in part B.